Imagine your body gets a little paper cut.
Right.
But instead of forming a simple scab and just moving on, it completely panics.
Oh, totally overreacts.
Right. It decides to just pour wet, unyielding concrete straight into your lungs, and it never stops pouring.
That's a terrifying thought.
It really is. So today, we're taking a deep dive into this massive, incredibly dense 2026 paper from the journal Cell Genomics by Jaiswal and colleagues. And, our mission today is to finally decode the cellular mechanics of how and why that runaway construction project actually happens.
Yeah, so we're looking at the molecular blueprints of interstitial lung disease, or ILD, and specifically its most aggressive form, which is idiopathic pulmonary fibrosis.
IPF, right.
Exactly. Yeah. Under normal circumstances, wound healing is a highly regulated, very transient process. Tissues get damaged. Local inflammation triggers cells to lay down a temporary structural patch made of extracellular matrix.
Like biological mortar.
Right, the mortar. And then the system dials back down and the tissue regenerates. But in ILD, the brakes on that entire system just fail completely.
They just snap.
Yeah, the inflammation persists. And the deposition of that extracellular matrix, it just becomes a continuous runaway loop.
Which is wild.
Right.
So to understand the sheer scale of the destruction here, I want you to think about the architecture of a healthy distal lung. It's fundamentally designed for maximum gas exchange.
It's extremely delicate.
Yeah, it's like an incredibly intricate, airy sponge. The walls of the alveoli, the tiny air sacs, they're lined primarily with alveolar type 1 or AT1 cells.
And those are remarkably thin, flat cells.
Exactly. So if the lung is building, the AT1 cells are like the ultra-thin glass in the window panes. They're designed to let oxygen pass right through into the bloodstream without any obstruction.
But, those delicate windows are constantly subjected to environmental stress. Pollutants, viruses, just general mechanical wear and tear.
Right.
So the lung relies really heavily on a second population of cells, the alveolar type 2 or AT2 cells, and these serve two distinct functions.
Okay, what's the first one?
First, they secrete surfactant, which is a specialized fluid that reduces surface tension so the air sacs don't simply collapse every time you exhale.
Crucial, obviously.
Right. And second, and this is critical to this paper, AT2 cells act as the resident stem cells.
They're the local window repairmen.
Exactly. When an AT1 cell is damaged and dies, an AT2 cell gets a signal, it divides, and it differentiates into a brand new AT1 cell to replace that broken glass.
Okay, so that is the healthy, functioning architectural plan. But to figure out why these window repairmen suddenly start participating in this runaway concrete pouring operation in ILD, the cell genomics researchers used a really cool piece of technology.
Single-nucleus RNA sequencing, or snRNA-seq.
Right, so they took flash frozen tissue samples from 36 subjects, isolating over 620,000 individual nuclei to read their transcriptomes. It's basically like putting a complex machine into a blender to get a complete parts list, right?
That's a great way to think about it. And the methodology here is vital. In deeply fibrotic, scarred tissue, trying to extract whole, intact cells is just incredibly difficult.
Because it's literally like concrete.
Exactly. The physical matrix is so stiff that the enzymatic processes you'd normally use to free the cells, well, they often just destroy them.
Oh, wow.
Yeah. So by targeting just the nucleus, the armored vault containing the RNA transcripts, they bypass that physical barrier entirely.
That's brilliant. It shows exactly which genes are actively being read.
Right. It allowed them to build a staggering high-resolution inventory of the disease. They could see exactly which cell types were depleted and which were hyperexpanded compared to a healthy lung.
And that inventory revealed an absolute biological disaster zone.
Yeah.
I mean, the structural foundation of the lung was collapsing.
A collapse.
There was a massive depletion of the AT1 window panes, the AP2 repairmen, and the delicate capillaries that normally surround the alveoli.
And filling that void was a massive expansion of highly pathogenic cell states.
The bad guys.
Right. The researchers flagged two major culprits. First, a heavily expanded population of fibroblasts labeled CTHRC1 high. And second, a mutated epithelial cell called an aberrant trait.
Aberrant transitional alveolar epithelial cell.
That's the one. Let's break down those labels because, the nomenclature really tells the story here. The high in CTHRC1 high just simply means these specific fibroblasts are expressing unusually high levels of a marker called CTHRC1.
And that marker is bad news, right?
Very bad news. It's heavily associated with aggressive, rapid collagen production.
So these are the rogue construction workers actively mixing the concrete.
Yes. Meanwhile, the aberrant TRAS-OS, they're essentially the AT2 repairmen who started the process of transforming into AT1 cells to fix a broken window, but they just got stuck halfway.
They stalled out.
Exactly. They stalled in this mutant intermediate state. They aren't functional stem cells anymore, but they aren't functional window panes either. They're just occupying space and driving disease.
Okay, so we are at our first mini summary. Let's do a quick recap for you before we dive deeper. We're looking at an ILD lung that's basically turning from a soft sponge into stiff concrete. The vital AT1 and AT2 cells are disappearing, and we have the single-nucleus RNA sequencing giving us this exact parts list. And that parts list shows us that rogue repairmen and hyper-aggressive fibroblasts are taking over.
That's the exact setup.
So here's a question for you to think about. Think about a time you tried to fix something in your house, but the repair process went rogue and actually made it worse. How do you stop a repair system that doesn't even know when to quit? That's the exact puzzle scientists are trying to solve here.
And, solving that puzzle required taking that massive parts list of 600 to 20,000 sequenced nuclei and actually putting it onto a physical map.
Because just knowing the ingredients doesn't tell you how the cake is baked.
Right. Knowing the ingredients of a disease doesn't tell you how it physically spreads through the tissue. So to map it, they utilized spatial transcriptomics, specifically the 10x Visium platform.
Let's actually pause right here for a quick 5-minute recap of the journey so far, just to lock in the bullet points.
Sounds good.
Bullet 1, ILD turns the soft lung sponge into stiff scar tissue. Bullet 2, we lose our functional AT1 and AT2 cells. And bullet 3, pro-fibrotic cells like those CTHRC1 high fibroblasts and aberrant trading cells multiply like crazy.
Perfect. So moving from the parts list to the blueprint.
The spatial map, it's like a GPS map, right?
Exactly like a GPS map. The spatial platform uses incredibly tiny 50-micrometer spots placed across intact slices of the lung tissue.
50 micrometers, that is tiny.
Super tiny. And each spot captures the RNA transcripts of the cells immediately touching it. Then the researchers computationally overlay this molecular data onto high-resolution microscopy images of that exact same tissue slice.
So they can see the actual geography.
Yes, they categorize areas into parenchyma P for the structurally preserved, healthy-looking zones, parenchyma F for the deeply fibrotic scarred zones, and then the transitional zones bridging the two.
And then they just drop the RNA data on top to see exactly which cells are living in which neighborhoods.
That's exactly how it works.
Okay, wait, hold on. Let me push back on this a little bit. If parenchyma F is the deeply fibrotic zone, the thick, dense structural scar, shouldn't that neighborhood be absolute ground zero for those CTHRC1 high fibroblasts?
You would think so.
Right. Shouldn't it just be packed wall to wall with the construction workers building the scar?
That's what makes this so fascinating. The spatial data revealed a deeply counterintuitive mechanism. The thickest, most heavily scarred regions of parenchyma F actually undergo severe decellularization.
Meaning there are no cells there.
Right. They become biological ghost towns. The active construction isn't happening in the center of the scar because the center is essentially dead, solid extracellular matrix.
Wow. So where is all the pathogenic action happening?
It happens entirely at the borders. The areas where the CTHRC1 high fibroblasts and the mutant aberrant tree cells are actively co-localizing and communicating.
Ah, the fibroblastic foci.
Yes, the fibroblastic foci. These are highly active, aggressive border zones where the healthy tissue meets the scar. The fibrosis acts like a slow-moving wildfire.
That's a great visual.
The leading edge of the fire, the foci, it consumes the healthy transitional and parenchyma P zones, and it leaves nothing but a dense cellular wasteland behind it as it expands outward.
It's just leaving a ghost town in its wake. And while that outward expansion is happening, the spatial mapping picked up another massive zoning violation, right? Something they call ectopic bronchialization.
Yes.
And ectopic basically means entirely in the wrong place. The researchers found airway cells, specifically basal cells and SCGB3A2 high cells, invading deep into the delicate alveolar air sacs.
Which is completely abnormal. Under healthy conditions, basal cells and SCGB3A2 expressing cells belong exclusively in the upper conducting airways.
Like the bronchi.
Right, the bronchi and larger bronchioles, the pipes that deliver air down to the alveoli. Finding them deep in the alveolar parenchyma is a complete collapse of anatomical boundaries.
Because the SCGB3A2 high cells are airway secretory cells, and the basal cells are airway stem cells.
Exactly. Their migration downward indicates that the lung is desperately trying to repair the dissolving alveolar structure by literally paving over it with tough upper airway tissue.
I look at this as the biological equivalent of a rogue highway. It's like looking at a map of a quiet residential neighborhood and finding that a massive, industrial toll booth has just been built right on top of a children's playground.
The zoning laws of the lung have completely failed.
Yeah, the architecture is just so distorted. Okay, time for our subtopic 2 mini summary. We've learned that spatial transcriptomics acts as our GPS. We found out that deep scars are actually ghost towns, not active construction sites.
Right, the action is at the borders.
Exactly. And airway cells are trespassing into the deep lung in this process called ectopic bronchiolization. So, listener, if you were mapping a city and saw industrial factories suddenly taking over a nature reserve, you'd know the zoning laws were totally broken. What happens to the communication between cells that causes these biological zoning laws to fail?
Well, that's the next big piece of the puzzle. The spatial map is simply the physical evidence of a complete signaling breakdown.
Okay, let's hit our 10-minute recap really fast before we break down the communication. Bullet 1, our GPS map of the lung shows active scarring happens at the edges of those ghost towns. And bullet 2, airway cells are trespassing into the gas exchange zones. Okay, so tell me about this signaling breakdown.
Sure, so cells govern their behavior and maintain their specific zones through chemical communication networks. They rely on ligands, which act like molecular keys, and receptors, which act like the locks.
Keys and locks, got it.
In a healthy lung, these networks are heavily compartmentalized. They are highly modular. The endothelial cells lining the blood vessels talk primarily to other vascular cells. The AT2 repairmen talk to the AT1 window panes.
But the cellular graph networks from this paper show that in an ILD lung, that modularity is just gone.
Poof. Vanished.
The distinct communication channels collapse into this chaotic open radio frequency. You've got alveolar cells mingling directly with systemic vascular cells and huge inflammatory immune clusters called lymphoid aggregates, and they are all just shouting over everybody.
The construction site is in total disarray. But buried in that chaotic network data is a really striking paradox.
Oh, I love this part.
Right. When the researchers ran centrality analyses, which are basically algorithms designed to measure which cell populations hold the most influence over the entire signaling network, the highest scores did not go to the hyperexpanded fibroblasts.
Which is what you'd expect. Yeah. But instead, the most central influential hubs were the AT1 and AT2 cells. The exact same cells that the single-nucleus RNA sequencing just proved were rapidly dying off.
Yes.
How does a cell population that is actively disappearing manage to dominate the entire communication network of the lung?
Because of the intensity of their distress signals. It operates much like a sinking ship or a catastrophic failure in a large construction site. Right. There might only be a handful of original site managers left, the remaining AT2 cells. But because the building is literally collapsing around them, those few remaining managers are screaming into their radios at maximum volume.
They're vastly outnumbered, but they are incredibly loud.
Exactly. Their distress signals are flooding the ligand receptor networks, pulling in inflammatory immune cells, and inadvertently signaling the fibroblasts to panic and pour more matrix.
But that raises a massive mechanistic question. We know these AT2 cells are screaming for help, but what is the physical trigger that forces them to actually mutate? How does a functioning stem cell stall out and become that pathogenic aberrant tree cell?
Well, the researchers couldn't answer that just by looking at the snRNA-seq data, or the spatial maps. They had to recreate the crime scene in a controlled environment.
Using in vitro human alveolar organoids.
Yes, organoids.
For those listening, organoids are essentially microscopic, simplified, three-dimensional versions of an organ grown in a laboratory dish. They're like mini lungs in a dish. They let scientists take a healthy human AT2 cell, isolate it, and then systematically introduce different variables to see exactly what makes it break.
And that's exactly what the team did. Through a process of graded evidence, they exposed the healthy AT2 cells to various combinations of the inflammatory signals that were dominating those broken communication networks.
And what did they find?
They discovered that the mutation isn't triggered by a single rogue chemical. It requires a highly specific trio of pro-inflammatory cytokines.
The three bosses of fibrosis.
Yes, TGF-β, IL-1β, and TNF-α. And the defining characteristic of this mechanism is synergy.
Right. Any one of these alone isn't enough.
Precisely. If you expose the AT2 organoid to just TGF-β, it doesn't mutate. If you expose it to TGF-β and TNF-α, it still doesn't happen.
So they have to hit together.
All three cytokines must hit the receptors of the AT2 cell simultaneously. They are jointly required. When that specific trio acts together, it forcefully overwrites the cell's genetic programming.
Whoa.
The AT2 cell abandons its normal repair function and locks into that stalled, maladaptive transitional state.
Okay, subtopic 3, mini summary time. The lung loses its anatomical borders, the depleted cells scream the loudest distress signals, and it takes a specific trio of inflammatory cytokines to mutate a healthy stem cell into a rogue transitional cell.
That's it in a nutshell.
So, listener, have you ever been in a situation where one stressful event was manageable, but three hitting you at the exact same time made you completely change your behavior? That is exactly what is happening to these cellular repairmen. They just get totally overwhelmed.
And once those epithelial repairmen break down, they drastically alter the microenvironment for the cells that actually build the scar.
Which brings us to our 15-minute recap. Bullet 1, cell communication becomes a chaotic, open radio channel. Bullet 2, the distress signals, our three bosses, force healthy cells to go rogue. So let's talk about the fibroblasts.
The CTHRC1 high myofibroblasts.
Right. We know the epithelial cells are going rogue, but who is actually pouring the concrete? It's the CTHRC1 high myofibroblasts. They are the construction workers. But what tells them to start pouring?
The spatial and single-cell data pointed the researchers toward a specific transcription factor called NFTC4.
NFATC4. A transcription factor is basically a master switch, right?
Exactly. They are specialized proteins that enter the nucleus, bind directly to specific sequences of DNA, and turn gene programs on or off.
So it's like a rogue architect issuing the final work orders to the construction crew.
Yes. NFATC4 is highly enriched in those specific CTHRC1 high fibroblasts. But identifying the architect is one thing. Proving they're the ones giving the orders requires literally removing them from the site.
Right, which they did using an experimental algorithm called Sereno knockdown on primary human lung fibroblasts.
Small interfering RNA.
I love this technique. Instead of trying to surgically remove the NFACC4 protein, researchers engineer synthetic RNA molecules designed to perfectly match the messenger RNA that carries the instructions to build NFATC4.
Right, so when the synthetic RNA binds to it, the cell's own defense mechanisms recognize it as abnormal and chop it up.
You're basically intercepting and shredding the architect's memos before they ever reach the construction crew.
Exactly. And when they utilized that knockdown pathway to silence NFAC4, the results were definitive. The massive induction of pro-fibrotic genes completely halted.
The construction crew dropped their tools.
They did. Without NFATC4, the fibroblasts couldn't differentiate into that hyper-aggressive state.
Yeah.
But here is where it gets really interesting.
Oh, my favorite phrase. What is it?
The mechanism of how NFATC4 is activated in the first place. It isn't just turned on by chemical ligands floating around. It is activated by mechanosensing.
Mechanosensing. The ability of a cell to literally feel the physical stiffness of its environment.
Yes. Cells interact with the extracellular matrix using surface receptors called integrins. They grip the matrix and pull against it to test its resistance.
Like pushing your hand against a soft down mattress versus a concrete wall. Your muscles just know the difference instantly.
Exactly. To test this, the researchers grew primary lung fibroblasts in vitro on specialized hydrogel substrates calibrated to exactly 0.5 kilopascals.
Which perfectly mimics the soft, airy compliance of a healthy lung.
Right. On that soft substrate, the fibroblasts remained relatively quiet. But as the researchers increased the stiffness to mimic fibrotic tissue, the mechanical tension acting on the cells physically triggered the activation of NFTCC4.
The physical tension turned on the master switch.
Yes. The protein moved into the nucleus and turned on the pro-fibrotic program, which means that as the lung gets slightly stiffer from initial damage, the fibroblasts physically feel that stiffness. That mechanical tension activates NFATC4, which orders them to pour more matrix, which makes the lung even stiffer, which activates more NFATC4.
It is a devastating, self-fulfilling mechanical loop. So subtopic 4, mini summary. CTHRC1 hyofibroblasts pour the concrete, but NFTC4 is the master switch that orders them to do it. Turning off NFTC4 halts the scarring process even when mechanical tension is trying to trigger it.
That's perfectly stated.
So bringing it all together to synthesize this entire deep dive, this 2026 paper represents a massive paradigm shift. They didn't just map a broken organ, they handed us an incredibly detailed hit list for future therapies.
We now know exactly what we need to target.
Right, we have the specific trio of cytokines to stop the epithelial stem cells from mutating, and we have the NFTC4 master switch to stop the fibroblasts.
But there is a cruel irony to lung fibrosis that this paper highlights. Because NFTC4 responds to mechanosensing, the very existence of the scar creates the mechanical tension necessary to create more scar.
It's a runaway train laying down its own tracks as it accelerates.
Right, and it forces us to bridge the gap between traditional molecular biology and structural engineering. These cells are responding to the laws of physical physics just as heavily as they are responding to chemical signaling.
Which leaves us with a highly provocative final thought for you to mull over today. If the physical stiffness of the tissue itself is signaling the body to continually create more scar tissue, how do we biologically trick the cells into thinking the tissue is soft again?
It's a huge question.
Think about how you might break a biological feedback loop that is triggered by physical physics. Could we alter those mechanosensing integrins on the outside of the cell to act like microscopic shock absorbers? It's a puzzle that bridges biology and engineering, and it's the next frontier in finding a cure.
It really is.
Thank you so much for joining us on this deep dive into the architecture of the lung. Keep questioning the mechanics of the world around you, and we will catch you on the next deep dive.